Hello, I'm Sue Romanoff from Edison Group. Thank you for joining this Paradigm Biopharma webinar. It's my pleasure to kick off this discussion about Paradigm 6-month data. Management will share their presentation and we'll follow that up with a brief Q&A. Today, we're joined by Paul Rennie, Managing Director and Chairman, Dr. Donna Skerrett, Chief Medical Officer, and Dr. Mukesh Ahuja, Global Head of OA. If you have any questions during this presentation, please feel free to submit them throughout the tab at the bottom of your Zoom panel. We'll address as many questions following management's presentations as we can. Paul, I'll hand that over to you here.
Thank you very much, Sue, and I'd like to thank Edison for the opportunity to present to shareholders today. I'd also like to thank shareholders and other interested parties for attending our presentation today. What I'd like to do at the start of this presentation, I realize that not everyone in the United States is familiar or as familiar with Paradigm Biopharma as our investors in Australia are. We have been on the Australian Stock Exchange since 2015, and we have many long-term investors who know the story very well. So what I'd like to do is just do a little recap and explain to you really the essence of what we're trying to communicate to you today. So first of all, the vast majority of the data that you'll see today will be around this phase 2 clinical study called Para-OA-008. Now, this is a phase 2 study that Paradigm initiated for a particular purpose, which I'll explain in a minute, but it initiated this while at the same time commencing its pivotal phase 3 clinical study, which is Para-002. Now, in relation to Para-008, which is a phase 2 study, it's a small study comprising 61 subjects, roughly 20 subjects per arm of either placebo, once a week pentosan polysulfate sodium, or twice a week pentosan polysulfate sodium. The primary endpoint of this study has been met. It was a successful outcome, which was reported in October 2022. So the study has reached its primary endpoint, which was day 56. So day 56, after the drug had been administered over a dosing period of 6 weeks, 2 weeks later, we did our primary analysis, and that primary endpoint was met. And we did a secondary follow-up, which was investigating the clinical outcomes of people on this study, and the same clinical outcomes are the primary endpoints for our pivotal phase 3 clinical study. That is change from baseline in both WOMAC pain and function. And that was statistically significant at day 56. So this study we're talking about today has been a successful phase 2 as defined by meeting its primary endpoint. However, we felt that given some of our earlier data from another phase 2b study that we conducted back in 2019, what we found was that there were significant signs or signals that this drug that we're talking about today may have some disease-modifying potential. So we wanted to, from an exploratory perspective, investigate whether this drug had disease-modifying signals. So even though some of the signals were unexpected by industry experts, we felt that they were very important signals. Some of the signals were strong enough to give statistical significance over placebo. Others showed very strong trends. So we put that into context of understanding that a study showing very positive signals trending to significance on 20 subjects is probably going to reach significance when you increase that study size to several hundred people. So just putting that into perspective, 78% of the people in this study were KL grade 3 or 4. So we're talking about people with moderate to severe osteoarthritis. And so it's obviously that's the group or the population with now quite severe pain and other morbidities associated with osteoarthritis. So it is a challenging group of subjects because they have established osteoarthritis and in significant pain. So with that as the background, I'd like to go to the next slide, please. As an ASX listed company, we are required by our rules and regulations to highlight the disclaimer and caution around forward-looking statements. Next slide, please. A little bit about Paradigm. As I mentioned earlier, on the left-hand side of the panel, you can see that Paradigm is an Australian public company founded in 2014, listed on the Australian Stock Exchange in 2015. Moving to the centre of the presentation, we can see that pentosan polysulfate sodium, the drug that we're talking about, which is a proven molecule, is administered to subjects in subcutaneous form. So we're not talking about intra-articular injection. We are talking about a subcutaneous injection, very similar to all of the monoclonal antibodies on the market today, like Humira, which as a subcutaneous injection can ultimately be turned into a self-administration, a product that can be self-administered. This particular drug, pentosan polysulfate sodium, or PPS, or iPPS as we refer to it, is a non-opioid drug with a 60-year track record history of treating pain, inflammation, and thrombosis in humans. The lead programs for Paradigm is investigating pentosan polysulfate sodium, or iPPS, or also trademarked xylitol, in the condition of osteoarthritis. So as I mentioned at the outset, xylitol is currently in a global phase 3 clinical study, and it's being studied with the primary endpoint of change in pain from baseline at day 56 for both WOMAC pain and function. The US FDA has granted this program fast track. We do have a globally harmonized protocol so that we can, post the phase 3, get simultaneous approval in all the key jurisdictions. So the same protocol is being accepted by the EMA, for example. So we are able to get simultaneous registration once the trial is finished. The drug administered subcutaneously is over a 6-week treatment course. And what we're going to talk about today in the Para-008 study, which is a phase 2 study, we're going to talk about the results that we're seeing at the 6-month time point. So bear in mind, these people have had a 6-week course of the drug, which is administered once a week or twice a week for 6 weeks. And now 6 months on, we're now seeing the results, and we want to talk to you about those today. Our second program is an orphan indication. It's currently in a phase 2 study in the ultra-rare disease of MPS 6. We've just finished a phase 2 study of MPS 1 in Australia. PPS has demonstrated potential to treat the residual pain that the MPS patients experience, albeit that they are on enzyme replacement therapy. So enzyme replacement therapy does have a lack of treating the residual pain for these patients. And so PPS, added in conjunction with the standard of care being enzyme replacement therapy, looks to attenuate their pain. So we see this as adjunctive therapy to standard of care being enzyme replacement therapy or bone marrow transplant. Through a number of phase 2 studies, Paradigm has been able to prove that we see meaningful treatment effects compared to placebo for pain, activities of daily living, joint stiffness, function, and PGIC. We have treated over 700 subjects in Australia under the federal government's special access scheme administered by the Therapeutic Goods Administration of Australia. And we have treated 10 subjects in the United States under the FDA's approved expanded access program. And we have collected these data. Although it's not a clinical study, it is still nonetheless very important clinical data representing real-world evidence of people who have osteoarthritis and have been treated with the drug. Next slide, please. Okay, so I'm going to touch on the highlights of this 6-month readout. Again, bear in mind, these people have received their drug 6 months ago, either once a week or twice a week for 6 weeks, and now we're looking at their outcomes at the 6-month time point. These are not pivotal endpoints. these are exploratory measures that we've undertaken with this phase 2 study. At day 56, the primary endpoint was achieved, as I mentioned, and it was also, the primary endpoint was a change in one synovial fluid biomarker compared to placebo. And Paradigm reported 7 favourable changes in synovial fluid biomarkers compared to placebo. So we actually saw disease markers going down in the PPS group and going up in placebo. We also got statistically significant improvements at day 56 in pain at 0.05 and at function at 0.017, and overall WOMAC scores for twice weekly iPPS compared to the placebo arm. So what is important here, I think, is that this is the same primary endpoint that we are using with the FDA, which is change from baseline at day 56 in WOMAC pain. And we've proven in this phase 2 study in a very small group of patients, only 20 per group, that we are getting a statistically significant readout at day 56. And that encourages us for our phase 3 study, obviously. Now, in addition to those outstanding outcomes, we have got multiple signals at day 168 or 6 months of disease-modifying efficacy of iPPS following a 6-week treatment. So they've had their treatment, now we're 6 months on, and we're seeing signals of disease modification. Objective measures of MRI at day 168 demonstrated changes in several structural features of the joint, which are consistent with disease-modifying efficacy. And most notably, we saw improvements in cartilage loss in the PPS group, whereas we saw advanced cartilage loss in the placebo group. Bone marrow lesions and marginal osteophytes were also reduced in the PPS groups, and they are very important markers for disease progression, the bone marrow lesions and osteophytes. Now, industry experts have told us that when we get to looking at biomarkers, synovial fluid biomarkers, the key 4 biomarkers that we should look at are these biomarkers of ARGS, C2C, COMP, and CTX2. Now, if you're not familiar with these, what these represent, these are pieces of the cartilage matrix. The ARGS is a piece of aggrecan, and when it's broken down by specific enzymes, they're broken into pieces. These pieces can be detected through analytical techniques in synovial fluid taken from the patient. So we have seen that all of these key biomarkers for disease modification, ARGS, C2C, COMP, and CTX2, all moved in the opposite direction to the placebo at 6 months. And some of our industry experts are wildly excited about these data. This has never been shown before in a controlled phase 2 clinical study. So this is a very exciting outcome. We also saw durable positive clinical responses in WOMAC pain, function, stiffness, and overall WOMAC score. And post day 56, we saw that rescue medication was used over 4 times more in the placebo group compared to twice weekly iPPS through to the 6-month timeframe. So we're seeing rescue medication being used in the placebo group at a much higher rate than the PPS treated population, which does introduce some positive bias for the placebo. But despite that, we haven't seen the line for the PPS pain moving back towards the placebo line. So we're actually quite excited to see, despite the increase in the rescue medication, which is paracetamol, very similar to acetaminophen, or NSAIDs, those drugs have not been able to drop placebo down to the same effect as PPS, albeit that PPS was administered 6 months ago. Next slide, please. Okay, so this is my final slide, and I'd like to just remind people, if they've heard the story before or people new to the story, why would a company be interested in pursuing a disease-modifying potential for its drug? I think the first and probably one of the most important reasons why a company would want to pursue that is that patients and doctors desperately want a drug that will give them symptomatic relief, so reduce their pain, improve their function, reduce the stiffness of the joint, but at the same time give them some hope that the disease is not going to progress to the end stage and therefore require a total knee replacement. I think many of us consider ourselves to be too young to be going in for total joint replacement. So there is a very strong demand from people who have osteoarthritis to stave off the disease as long as possible. And we also know doctors feel the same way. They want a drug that's safe and effective and can also possibly reverse or slow down the progression to end-stage disease. And it's important that people understand that currently there are no approved disease-modifying osteoarthritis drugs for osteoarthritis. We also know that 81% of OA patients are dissatisfied with the current OA therapies, and that was from a peer-reviewed publication. And Paradigm commissioned its own global market research in 2021 and spoke specifically to payers around what the impact of disease-modifying label for iPPS and reimbursement would be. And so what we took from that research was that if there was a disease-modifying label, then this drug would probably move from second-line therapy into the treatment algorithm to first-line therapy, and therefore be used more regularly and increase the overall demand for the product. We also know that reimbursement agencies would consider disease-modifying label as being very positive in terms of pharmacoeconomics and staving off a joint replacement, and therefore attract a higher reimbursement price. The prices that we were quoted was for a pain and function label only of around $2,500 per course of treatment. So that's 6 or 12 injections over that 6-week period, or disease-modifying label where the price would then move to $6,000 per course of injection. So it makes a significant and profound impact on the commercial opportunity here. And it's not the only reason why we're pursuing it, but obviously from an investor's point of view, there is a blockbuster opportunity here in the treatment of osteoarthritis for safe and effective drugs, even if it's just for treatment of pain. But when you move into this disease-modifying space, the numbers just get incredibly large. And I think, you know, as an industry, we've seen what's happened with some disease-modifying drugs, such as the anti-TNF therapy used for treatment of disease-modifying in a rheumatoid arthritis situation, rheumatoid arthritis being only 1/10 the incidence of osteoarthritis. But having said that, we know that Humira, over the last 20 years, since it first came onto the market, has generated over $200 billion because it's moved from symptomatic treatment in rheumatoid arthritis to disease modification. So disease modification completely shifts the paradigm to a different set of numbers. And in the case of osteoarthritis, we're talking about possibly shifting it into, you know, $10 billion per annum, up to $20 billion per annum. And those figures are not unreasonable, given that that's the sort of average figure that AbbVie got from Humira over the 20-year period. So that's my introduction, and I'll now pass over to my colleague, Dr. Donna Skerrett, who is Paradigm's Chief Medical Officer based in New York. Thank you, Donna.
Thank you, Paul. So now I'd like to go into a little further detail around the disease-modifying aspects of the programs that Paul has mentioned to you. As he mentioned previously, we conducted a study, Para-005. It's a completed study. It was 126 participants randomized to PPS or placebo. The dosage was 2 milligrams per kilogram twice weekly for 6 weeks versus placebo. And in addition to the very nice clinical responses that we saw in that study, we did get some signals about biomarkers and MRI. And that is that with the biomarkers, we saw reductions in serum levels of COMP as well as Adams TS5, and a reduction in the urinary levels of CTX2. And these are all markers of cartilage degradation. So seeing a reduction in these suggests that we have some preservation of the cartilage in the PPS treated arm compared to the placebo arm. On the MRIs, these patients had bone marrow edema lesions at entry into study. And the follow-up MRIs indicated that we saw clinically meaningful reduction in the PPS group compared to placebo. So these were two very nice signals regarding biomarkers and MRI findings suggesting disease-modifying potential for PPS in OA. So as you've heard, we then initiated this study, Para-OA-008, which is ongoing in terms of the 12-month follow-up. We've randomized 61 participants into this study, and they received a dose of 2 milligrams per kg once weekly, twice weekly, or placebo. And as you've heard, the primary endpoint here was to achieve some signals in the synovial fluid biomarkers. Why were these a focus of this study? In the prior study, we showed that we could measure serum biomarkers that gave us clues about cartilage preservation. Given that this drug is delivered by a subcutaneous injection, we wanted to get some evidence of the effects of this drug in the knee itself, and that was done by examining the synovial fluid. So at day 56, we saw signals from several biomarkers in the synovial fluid. We saw reductions in inflammatory cytokines TNF and IL-6. We saw reduction in the pain mediator NGF. We also saw a reduction in byproducts of cartilage degradation COMP and ARBs, and an increase in an endogenous inhibitor of cartilage degrading enzymes, and that's TIMP1. So again, we're now looking at synovial fluid biomarkers, seeing evidence of this at day 56, which is the same time when we evaluate our clinical endpoint for our primary endpoint for our phase 3 program as well. Next slide. So these findings are consistent with our understanding of the mechanisms of action of PPS. We know that primarily through NF-kappa B, we have effects on inflammatory pathways, pain through NGF, tissue preservation through a number of pathways, and as well as improved local circulation, which may be aiding in the reduction of bone marrow edema lesions. Next slide. So more on the findings at day 168 on Para-008. This biomarker study wanted to look at changes from baseline in multiple objective measures associated with disease progression of OA. I mentioned we have 61 participants receiving once or twice weekly dosing or placebo. And the follow-up period out to this of the study is out to 12 months. So we're now at reporting data at 6 months and have previously reported data at day 56. At day 56, we did see multiple biomarkers that provided insight into the mechanisms and disease-modifying potential. We also showed at day 56 that we had statistically significant improvement in WOMAC pain, function, stiffness, and overall WOMAC score for the twice-weekly arm. And so this is the endpoint that would be comparable to our phase 3 program endpoint. We saw significant changes in pain and function. We noted that significant changes in pain and function were not apparent in the once-weekly iPPS group compared to placebo. So the strongest clinical findings were seen in this twice-weekly treatment group. Next slide. In terms of trying to understand what's going on structurally, the tools that are most informative to us are the MRI examination of the knee. And that's because with MRI, you can get greater detailed information around bone marrow lesions, joint synovitis, cartilage thickness, bone shape, including osteophytes, and joint space width. So whereas traditionally an X-ray was used to measure joint space width, we're able to get much more information, including joint space width, by looking at MRI. And the findings in these various regions of the bone will correlate to certain types of pathology in the bone, and those are listed on the right column of this slide as well. The findings in the day 168 assessment by MRI provided us with signals that were often difficult to detect as early as 6 months. Very often we're advised that it's more likely that we will see changes at 12 months, but we were very pleased to see that there were measurable changes in the MRI findings at 6 months. And for further description of those findings, I will pass on to Dr. Mukesh Ahuja to describe the MRI findings. Next slide, please.
Thank you, Donna. Welcome everyone for joining Paradigm and for us to share with you the results of the Para-OA008 study. So here I will present the day 168 top-line data from the MRI-based semi-quantitative scoring system called WORMS. So WORMS is an abbreviation for whole organ magnetic resonance score, which is a comprehensive scoring system used extensively in OA studies worldwide. The MRI images were read by an independent organization with trained musculoskeletal radiologists for accurate and reliable reading. The MRI images were obtained at 2 different time points. First, at the screening visit, at which pre-treatment OA disease characteristics were established, and then follow-up MRIs were obtained at day 168, which is 6-month follow-up, to identify differences in disease progression between the iPPS and placebo group. It is important to note here that despite the relatively small number of subjects in each arm of this study and the short follow-up interval, compared to the generally slow structural progression in osteoarthritis, changes consistent with D-mode efficacy were observed in a number of disease features. These structural changes were most noticeable in cartilage, bone marrow lesions, and osteophytes. With the next slide, I will share key findings for these 3 disease features. The first one is cartilage loss, which is known to be the most important disease feature for osteoarthritis. It's also considered the joint space width, which is a surrogate for cartilage loss. So with this WORMS scoring system, we have directly looked at the cartilage loss score. It's also considered to be predictive and clinically relevant endpoint for knee replacement surgery. In the Para-OA008 study, once-weekly iPPS cohort showed an average improvement of 21% in mean cartilage loss score in the medial femur, whereas the placebo arm showed a 4% worsening of cartilage loss. The twice-weekly iPPS group showed stabilization of cartilage compared to the placebo. The next disease feature with a statistically informative change is bone marrow lesions. Bone marrow lesions are related to microtrauma resulting from overloading caused by loss of normal bearing function of articular cartilage. Numerous studies have suggested bone marrow lesions are also predictive of knee replacement. In this study, bone marrow lesions in the lateral femur decreased by an average 38% in the once-weekly iPPS arm, whereas in the placebo arm, it increased by 47%. In the entire lateral tibiofemoral compartment, bone marrow lesions decreased by an average 17% in the once-weekly iPPS arm, whereas increased by 56% in the placebo arm. Twice-weekly iPPS group showed improvement compared to placebo. Next slide, please. Marginal osteophytes, also known as bone spurs. These are osseous outgrowths and is a common feature of osteoarthritis. They are believed to form as an adaptive response to mechanical stimulation or inflammatory cytokines, according to more recent research. The osteophytes increase in number and size as the OA disease progresses. As shown in this figure, with the increase in Kalgren-Lawrence osteoarthritis grading, increase in severity of osteophytes is seen. So in this study, osteophytes decreased or remained stable in all three compartments of the knee in patients treated with iPPS compared to an increase in the placebo arm. With that, I will yield back to Dr. Donna Skerrett to go over clinical and molecular biomarker results and also discuss about the next steps.
Thank you, Mukesh. So at day 168, the following clinical changes were observed. The twice-weekly iPPS group demonstrated durable responses in WOMAC scores for pain, function, stiffness, and overall WOMAC index compared to placebo. Specifically, we saw durable WOMAC pain reduction. We saw WOMAC function achieve 50% improvement, or 53.3% of the twice-weekly cohort compared to 22% in the placebo group. We saw PGIC was favorable at day 168 with a strong trend. And we saw that at day 112, iPPS showed WOMAC stiffness with significant function was a high trend, and overall also a very high trend in terms of improvement for the PPS group compared to placebo. Importantly, the placebo group used rescue medications 4 times as often as the twice-weekly iPPS group. So in other words, the PPS group was improving in pain and therefore improving in function. But with that improvement in function, we didn't see a recurrence of pain requiring them to start taking medication. So we've reduced medication use in the PPS group by about 4-fold. Next slide. This is an overview of the molecular biomarkers that have potential as identifying disease-modifying aspects for PPS for OA. We've spoken to the first 2 categories previously, where at day 56 we showed favorable changes in pro-inflammatory cytokine reduction and also reduction in the pain mediator nerve growth factor. And previously, we've seen some changes in the serum in para-005, but many more biomarkers were explored in this study in order to understand relationships between serum, synovial fluid, and urine biomarkers, and to also understand if we were seeing consistent and concordance changes in biomarkers suggesting disease-modifying potential. Next slide, please. So the day 168 biomarker results include evaluation of synovial fluid, serum, and urine biomarkers. And the iPPS groups identified disease-modifying potential in the OA treatment as demonstrated by alterations in 4 of the biomarkers. These 4 biomarkers are all related in that they all are connected to cartilage breakdown, and therefore the identification of these as concordant with the mechanism of cartilage preservation. Synovial fluid and serum samples of ARDS and COMP show favorable changes for PPS compared to placebo. And the serum C2C and urinary CTX2 also demonstrated beneficial effects for PPS compared to placebo. And next slide, please. And just to give you an idea of the concordance between serum and synovial fluid biomarkers, we wanted to show that C2C in the serum was reduced, and that reduction was statistically significant compared to placebo. For CTX, the finding was reduced. For COMP in the serum and the synovial fluid, both of these were reduced compared to placebo. And similarly for ARDS, the synovial fluid and the serum markers were reduced compared to placebo, with the synovial fluid reduction reaching statistical significance at 0.024. So again, these are all biomarkers of cartilage degradation. where iPPS treated subjects showed favorable profile compared to placebo control, thus providing us with a concordant profile of biomarkers to use when assessing disease modifying activity of PPS for acquiring additional data in support of a D-mode label. Next slide, please. So in terms of what our next steps are, we have acquired a lot of new data. We have MRI, molecular biomarker and clinical outcome data, which will be presented to the regulatory authorities, the primary meetings planned for the FDA and EMA. And we will have discussions with these groups during the second half of this year in order to understand what additional data and how those data should be evaluated and analyzed in our larger controlled study, PARA-005, in order to provide data that would be acceptable for obtaining a disease-modifying label language, in addition to the primary endpoint label for our phase 3 study, which is pain and function. This activity is not expected to impact the paradigm clinical timelines for OA. As I mentioned, we will have these regulatory discussions within the coming months in order to include the biomarker endpoints in the design of the confirmatory study PARA-003. We have previously announced that this program has fast-track designation with the FDA, and that facilitates easier access to the agency and an opportunity for frequent dialogue on the development program. Feedback from EMA will also be useful to us in terms of our next steps in with approaching the TGA regarding their provisional approval program. The data set from day 56 and now day 168 time points in PARA-008 will be prepared for peer review and publication. We have acquired a large data set now of molecular biomarker data, including synovial fluid, serum, and urine biomarkers. We have acquired now MRI data, which is showing changes at 6 months in PPS compared to placebo subjects. And these are both areas of great interest to the osteoarthritis community and to those who are specifically interested in identifying biomarkers as surrogates of response for OA development. Next slide, please. I'd now like to say a quick word about the phase 3 program. I've mentioned that we've conducted and completed a phase 2 program, 005. The data we presented to you today is from 008. 6-month data are available now, although these patients will be followed out to 12 months, so there will be additional data on them. We are currently enrolling in OA-002, which is our pivotal study. It has 2 stages, and the dose finding stage, which is evaluating 3 different doses compared to placebo, is expected to complete enrollment by the end of June this year. Following dose selection, we will then move into our confirmatory phase 3 study, 003. So we'll have data from 2 phase 3 studies as we go into registration submissions. Both of our phase 3 studies have extension arms, so we will follow patients out to 12 months in order to acquire duration of effect data. And given that OA is a chronic disease, and our feedback from many patients who have received PPS through the SAS program in Australia, we will use our duration of effect data to design a retreatment protocol in order to have controlled data on retreatment responses. And we also, as we've previously mentioned, will initiate a program for OA of the hip, since it has a similar regulatory pathway for OA of the knee, and that will be developed once we have the dose selection complete. So I'll now move to the next slide and hand over to Paul for wrap up.
Thanks very much, Simon. And Sue, I just finish off this last slide, which is the near-term news flow for investors, so that I can then hand over back to you. As I mentioned at the outset, Paradigm has a number of programs. We have the osteoarthritis program, our lead program, but we also have a program in phase 2 for the ultra-rare disease of MPS 6. And we'll be announcing shortly the finalisation of the recruitment of that study, which will be in the second quarter of this calendar year. So that's imminent. As we're running our phase 2 and phase 3 studies, as you've heard, we're also running a canine OA model. Our scientists tell us that the canine OA model, being the natural osteoarthritis, not an experimentally induced osteoarthritis, very nicely translates into a similar outcome in humans. So it is a natural history OA model, and the advantage of looking at disease biomarkers in canines is that they age much quicker than humans, so therefore a 26 follow-up in canines is equivalent to a 3-year equivalent in humans. So that data will be out before the end of June this calendar year, and we are seeing some very interesting outcomes from that study. Para-002, which is our pivotal phase 3 clinical study, will be updating recruitment on that towards the end of Q2 this calendar year. Of course, as we've just heard, Para-008, which is a phase 2 study, we discussed that the study was successful. It met its endpoints at day 56, but exploratory work showed that we do have very significant signals of disease modification at 6 months, and we'll be reporting the 12-month follow-up from all of those patients in the second half of this calendar year 2023. We will be also presenting the MPS 1, which was a phase 2 study conducted in Australia, and the MPS 6 phase 2 clinical studies. We'll be reporting those top-line data at the end of Q4 this calendar year 2023. And Paradigm is currently in active discussions with multiple potential partners for its phase 2 asset, the orphan indication of mucopolysaccharidosis. And that continues to progress and become very interesting as we just had the World Congress meeting in Orlando, Florida. We had a lot of interest from investigators inquiring about the trial, a lot of people from MPS societies very interested, because this is one of very few double-blind placebo-controlled study in an ultra-rare disease. Most ultra-rare diseases are open-label studies, but this is double-blind placebo-controlled study. So that is the end of our presentation. So I'm going to hand back to Sue, who will now forward some of the questions that have been received throughout the presentation. So back to you, Sue.
Thank you, Paul. We have a lot of questions here today. If we don't get to your questions, we'll do our best to follow up with you directly. The first question has 3 parts. Here's the first part. Could management please comment on the differentiation in the cartilage degeneration data in the once-weekly group versus the twice-weekly group?
I can, I'll take that question.
Mm-hmm.
So yeah, overall the cartilage score, we use, as I explained earlier, we use Worms cartilage loss score. So overall the score was stable in twice weekly group and improved in once weekly as compared to placebo, which means there was no meaningful cartilage degradation identified in twice weekly, and possibly, it's a signal that there might be regeneration in once weekly group. So I'm cautiously optimistic about it, but the improvement in cartilage loss score in once weekly was very encouraging, and that in twice weekly, the cartilage score was still stable.
Okay, the second one, could the twice weekly group have a better reduction in pain and thus been more mobile than the first, the once a week group, causing faster cartilage degeneration?
So,
I'll take that question as well. So our answer to the first part of question is likely yes. And for the second part, we did not find any evidence of faster cartilage degradation in twice weekly.
Mukesh, one quick last follow-up to that. Considering the difference in the once weekly group better than the disease modifying results versus twice weekly group better pain function, will this cause problem in choosing the dose scheduling going forward?
No, we don't think so. The dose selection is based on pain function and safety data up to day 84.
Okay. Paul, maybe this is for you. Where do we expect iPPS to be launched first?
well, I think iPPS will probably be launched first in the United States. Obviously, the United States is the world's largest market. We are doing our pivotal phase 3 study in the United States. So that's our plan. I think timing, as you probably saw from the slide from Dr. Skerrett, is we'll be submitting our request for registration via an NDA to the US FDA towards the end of 2025. And we would hope that first revenues would occur within the month after that submission. I should also just say that due to the way that this study has been designed, if we continue to explore disease modification and have a disease modification label, that will not extend the time to revenues. The disease modification studies can be done within the phase 3 studies that are either ongoing or about to commence. So the pivotal study is ongoing, and we can, at the end of the dose selection, incorporate changes in the protocol to collect disease modification data for a disease modification label, or we can get the disease modification data from the confirmatory study, which is Para-003, and that will commence after the dose selection in Para-002.
Donna, this one might be for you here. How remarkable is the improvement in cartilage? Has there ever been achieved in humans?
The concordance between improvement in MRI as well as the molecular changes seen in the biomarkers with clinical improvement is particularly unique to this program. So we consider these very strong signals for disease modification along with clinical effects for PPS. So this is, always we're cautious because we will be acquiring more data, but we consider this really remarkable findings to see, especially at 6 months. Yeah.
So
Okay.
I might just, sorry, Sue, could I just jump
Yeah.
in there? I completely agree with Donna, and one of the things that I do is obviously from a commercial perspective, keep an eye on what is out there in the market. And I'd have to say at this point in time, there is no previous study that has actually shown improvements in cartilage to the extent that we've shown in this particular study. So I think it is really a world first and quite groundbreaking. The other thing I'd like to mention is that in addition to cartilage, I don't think that there has ever been a drug registered where the subject is dosed and 6 months later, the effect is still significant. So I think that's another important aspect of this drug. You do the administration and you get long-term durable effect from the symptoms of pain and joint stiffness. So I think it really is quite a unique outcome of the data that's been seen at 6 months.
When do you anticipate having enough information to know the dosage you'll use throughout the larger upcoming 002 and 003?
That's planned to occur as part of an interim analysis for the first stage of Para 002, which is enrolling now. The schedule for completing enrollment is by end of June this year. And so within a few months following that, we should have our dose selection completed.
Okay. The potential to maximize the impact of iPPS by demonstrating a disease-modifying profile in the KOA in phase 2 Para-OA-008 trial. It's very exciting. And then the 6-month data is particularly encouraging. What do you believe are the most important outcomes for the 12-month data? And what do you believe the FDA and EMA will need for the disease-modifying indication labeling?
Sure, I'll comment to that. So traditionally, disease modification has required evidence of structural preservation. Initially, that was determined by joint space narrowing. More recently, some of the other findings, particularly those around preservation of cartilage, have been reviewed by advisory groups working with the FDA. So when we think about the difference between 6 months and 12 months, it would be ideal to see the persistence of these favorable structural changes. The reason we have interest in some of these structural changes is because some of them have been correlated with deferring or the need for a total knee replacement. So when you think about the long-term clinical endpoint, what sort of structural evidence can we provide that will be sort of a surrogate for that longer-term outcome of total knee replacement, which is really more challenging to prove in the clinic. So I would say certainly to see persistence in the structural changes, also perhaps to be able to correlate some of the molecular biomarker findings with changes in structure as well, because again, we have clinical evidence, we see that it's durable. It would be good to have an understanding of, you know, if these, the biomarkers that are showing us some preservation of cartilage and slowing down that degradation activity, what does that mean later down the line in terms of the structural findings? So all of these will be evaluated by our statistical group.
Okay, in reference to the pivotal PARA-OA-002 phase 3 trial to assess the long-term efficacy and safety of iPPS in patients with KOA pain, is patient enrollment progressing as planned? And can we expect to hear an update from the study?
I would say Mukesh, who is the champion of the enrollment, should speak to that.
Yeah, thank you, Donna. So yeah, we are very excited. We have about close to 100 sites activated globally on this study, and the enrollment is going rapidly, and we have pretty much doubled up our recruitment rate in the last 2.5 months. So yeah, we are very excited about the recruitment pace for 002 study.
Okay, maybe a couple more. Is the preparation for confirmatory phase 3 Para-OA-003 trial progressing as anticipated? And are you still planning to initiate this in calendar year 23?
Mukesh, do you want to speak to that, or?
Yeah, so for 003 study, 003 study is planned to be initiated once we have selected the dose from stage 1 of 002. that's when we will also initiate our phase 3 confirmatory study 03, which is expected to happen sometime later this year.
Okay. And the treatment para-OA 09 trial expected to initiate in calendar 24 to assess the feasibility of multiple treatment regimens for iPPS. Do you anticipate the results from the study will have significant influence on the company's clinical development plans for iPPS?
I'll take that. I don't know that it will have a significant impact on the development plan. The development plan is pretty much laid out before you, as you saw on that timeline. What this will provide for us are controlled data around retreatment. And we have experience with several hundred patients in the SAS program in Australia. So we know from those patients that they come back sometimes a year and a half, saying they are sensing that the effects of their initial treatment are wearing off. Can they be considered for retreatment? We would need to document that in a controlled study, which is what we're doing with the extension study data. And then around that, design what retreatment should look like for a trial, just to put some retreatment data in front of the agency. But I don't see that having an impact on the development plan, major impact, yeah.
Okay, maybe this last one here, period calendar 23-25, it looks like it's particularly active. What's the company's strategic priorities for this period, and what do you believe are the most significant near-term catalysts for investors to look for?
Well, I might take that question. Thanks, Sue. So in terms of near-term catalysts, I'm not sure if they still appear on the screen. I can see them on my screen, but essentially what they are is, if we sort of generalize them, it means readout of the orphan indication top-line data towards the end of this calendar year. We also see further readout of this particular study, 008, at the 12-month follow-up from all of these subjects. We'll also have an update on the pivotal study towards the end of June, an update on recruitment rates and any news thereafter about dose selection. And the other piece of information would be mainly possibility of some sort of commercial transaction around a licensing of the orphan indication to a company interested in developing orphan treatments for orphan diseases. So we do have quite a number of commercial companies interested in both the MPS asset as well as the osteoarthritis asset. And I think that the data that we've just seen, again, noting that 008 did meet its primary endpoint, we're encouraged that that endpoint is the same as the pivotal and confirmatory study, change in pain from baseline at day 56. We met that with statistical significance on a very small number of subjects. We would hope that that would give investors confidence that the same endpoint in a much larger group of patients would be a positive outcome. But having said that, I think what we're really looking for is that, as Dr. Skerrett said, maybe a continuation of the structural improvement at the 12-month endpoint. And if that's the case, then I think, that would be the point for us to be able to really come to a value on this asset, to be able to establish an equitable financial transaction in relation to the osteoarthritis asset.
We're going to squeeze one more in. I know I said that the last couple of times. Is exercise undertaken by patients during the study controlled or tracked for the 3 groups? From your learnings in the study, is this something that you would build in the confirmatory 003 study?
If I can address that with regards to the design of the phase 3 program, patients may exercise, of course. They are asked to refrain from initiating any new, very aggressive programs. So, for example, if they're doing physical therapy, to continue with that. We do track that activity. It is important to track that as it is also important to track all the medications that are used. But as patients feel better, they do want to do more, and so we do keep data around how active they are.
Okay, great. That's really all the time we have for questions here today. Thank you everyone for joining here. If you have any other additional questions or we didn't get to your questions, or if you'd like to speak to the team one-on-one, please reach out to the contact information listed on the Paradigm website. Thank you.